Hi everyone, and welcome to today's lecture, which is called Learning Through Data Mining on Open Datasets. My name is Emily Finn, and I'm a postdoctoral fellow in the section on functional imaging methods at the National Institute of Mental Health. And I've benefited greatly from open datasets over the course of my career so far, so colleagues and I have managed to gain some pretty interesting insights on brain behavior relationships using these types of data. So my goal today is just to give you a conceptual overview and share with you some of the insights that I've gained over these last few years of working with this type of data. So a few notes about today's talk before we get started. I'll be focusing mostly on what I'll call large open data sets, which I'll abbreviate LODs, and how these can fit into and further the cause of reproducible science. So what I mean by large open data set is typically a data set with hundreds of thousands of subjects with a large number of both imaging and behavioral measurements on each subject. And it's important to note that with the advent of online data sharing platforms, things like NeuroVault or OpenFMRI, which is now OpenNeuro, hopefully someday soon this will be a little bit of an oxymoron because every fMRI data set, no matter how big or small, will be an open data set, and certainly there will be a lot to be learned from aggregating and reanalyzing some of the smaller and more focused data sets. But just to avoid confusion, for today's purposes, I'll mostly be talking about larger data sets, which I'll abbreviate LODs, and you've probably heard of many of these. These would be things like, for example, the Human Connectome Project. And as a disclaimer, there'll be a pretty strong focus on individual differences today, since that's my area of interest, but I also think that it's the place where these types of large open data sets can help us make the most progress. There will also be a functional connectivity bent, since many large open data sets contain mostly or exclusively resting state data as their functional data, since resting state is particularly easy to acquire and share. But hopefully the concepts and the principles that we'll discuss today should be applicable to a wide variety of research questions and analysis techniques. So part of what I'll talk about is ways to hopefully inspire you to see how you might be able to use a large open data set either on its own or in combination with your own smaller lab-grown data sets. And throughout and towards the end, I'll also talk about some pros and cons of working with large open data sets and just things to be aware of before you dive in. And whenever I talk about a data set, I'll always try and provide a link to it, since the best way for you to learn about these data sets is just to go directly to the source and explore the richness of the imaging and behavioral measurements that are available for these large numbers of subjects. So when you see this little red arrow, that is a link to the data set itself that you can go and explore. Okay, so to place this particular talk within the broader context of this course, I think people mean a lot of different things when they talk about doing reproducible science. So at a very bare minimum, we could think about an analysis that's repeatable. So could you or someone else in your lab use the exact same data and the exact same code to get the exact same output? So if you run an analysis today, can you shut down your computer, come back in tomorrow, turn it back on, run the exact same script and get the exact same numbers? Or could you, ship your data and your code to your lab mate and have them get the exact same numbers? And we hope that the answer here is yes. That's sort of, again, our bare minimum threshold. But, in the fMRI data sharing world, we'd like to start moving beyond this bare minimum towards reproducible results or analyses. And what this term has come to mean for many of us is this question of, can someone in a different lab use the same data and the same code to get the same output? So if you stumble across a cool new paper and you see a figure that they've created, is there a place that you can go to get both their data and their code and actually run it yourself to reproduce that exact same output? That's what some of these cool new tools are helping us to achieve, and these will be covered expertly by other speakers in this course. But our focus today is sort of a little bit more abstract than either of these, and that's on something that we'll call robustness or generalizability. So in a more conceptual way, given that you've discovered something about the brain in a certain sample using a certain type of analysis methodology, would it be possible for you or someone in a different lab to actually use different data and maybe, similar but not exactly the same code to get a similar output? So how robust is your finding to these things like which subjects you use or exactly how you implemented your question and things like that? And that This piece of things is what I'll focus on today because I think this is where large open data sets are uniquely positioned to help us start to answer these types of questions. And just as a quick note, if you're interested in learning more about this, I recommend this article that came out earlier this year in Nature Neuroscience, Best Practices in Data Analysis and Sharing in Neuroimaging Using MRI. And we'll be focusing down here. The authors provide a partial taxonomy of these reproducibility terms. And we'll be focusing down here on this generalization over materials and methods. So talking about things like near replicability, meaning if you do the exact same thing in a different pool of subjects, you get the same result. So large open data sets can help us with that. Because they're so large, we can do things like cross-validation to try and answer this question. But there are also sort of these other levels, so intermediate replicability, meaning can different labs run the same experiment and get the same results. Things like far replicability, where you're varying the experimental and analytical methods, and the holy grail of hypothesis generalizability, meaning you're looking not only you're changing as many things as you can to try and break your result, and you're seeing if that concept can still hold. And I think these last three things are things that can be addressed by either combining multiple large open data sets or combining a single large open data set with smaller lab-grown data sets, etc. So I'll give some more specific examples for how we can start to use large open data sets to answer these types of questions. But before I go any further, I'd like to motivate my own focus today on individual differences by just explaining a little bit of what I mean by that. So I think it's really important to not only look at things at the group level, but look at them on an individual brain to individual behavior scale. And from a basic neuroscience perspective, I think this mapping is going to help us gain a lot more traction and get us closer to, I won't say a mechanistic understanding. fMRI is never going to be a causal manipulation tool, but it does help us sort of bring out more subtle, more rigorous findings in our data. And of course, if we're hoping that we can eventually make tools, fMRI-based tools that are useful clinically, we'll need a way to move sort of away from these categorical approaches where we scan 20 controls and 20 patients and see what's different between them, to more dimensional approaches where we scan a bunch of individuals and measure something about each person uniquely and sort of place everyone on the same axis and try and relate brain variables to phenotypic variables in this more continuous way. So I will argue that large open data sets give us not only the opportunity, but actually the responsibility to look for and discover brain behavior relationships at the individual level that are maximally robust and generalizable. The reason we have the responsibility to do this is that this is the only way that we're ever going to be able to develop tools with translational utility. So before I go any further, this is just sort of an advertisement slide for the many different large open data sets that are available out there. This is just a slide to illustrate some of the better known ones. It's by no means a comprehensive slide, but I have limited this list to data sets that include both functional brain data as well as some kind of behavioral or phenotypic measurement. So if you're looking for somewhere to get started, any of these would be a great bet. I won't go through them in detail since you're better served by just going to the project websites themselves and exploring. But you can see these all have a pretty large N, which is very nice. That's kind of the idea here. One thing to note is that some of these are prospective designs, like the Human Connectome Project, where there's a single grant to a single institution to prospectively collect a lot of people under the same protocol. And others of these are retrospective aggregations of data. So things like the ADHD 200 Consortium or the ABIDE datasets are actually made up of a lot of smaller datasets from different sites that were aggregated together in a post hoc way. And it's important to note that just because data were collected at different sites, it's not necessarily a weakness of the data set. In fact, it could be a great opportunity to actually test some of those questions of generalizability that we were talking about before. It's just something to be aware of when you're selecting one of these data sets to begin working with. Okay, so what types of things can you do with a large open data set? Say you've never even worked in neuroimaging. Well, guess what? You're lucky because there's no longer a need to collect your own data. You can actually just go online and download a bunch of fairly high quality data that someone else has collected. And what kinds of things can you do with this data? Well, I think the nice thing about these large sample sizes is that they allow us to be a lot more rigorous in our statistics. So they allow us to try to discover a brain behavior relationship in one sample and then immediately see if it generalizes to a new sample, without having to go through months or years of extra data collection, at least initially. And this is something we call cross-validation. You're probably familiar with this. But just as a very general overview of how this would work, we have this nice large N here, lots and lots of people that we've scanned. What we can do before we even start any kind of analysis is we can set aside a chunk of this data as the test set. So we're not going to look at this yet. We're going to proceed with all of the subjects in our training set. We're going to set aside our test set. And we're going to bring it back in later on to see how generalizable whatever result we discovered in our training set really is. And there's a couple ways we can do this. So harkening back to those examples from a couple slides ago, if we have a categorical variable where we're interested in whether someone is a healthy control or a person with a psychiatric illness, we can train some kind of classifier. This is just a really simple representation. So again, instead of just sort of looking for this difference in our training set and then, washing our hands and going home and saying we found a statistically significant difference, the fact that we've left out some data in this test set here means that we can then bring individuals back in and test to see how robust this result really is. So can we predict in this new test set whether a person is a control or a patient? And that's going to tell us a lot more about how useful our result really is. Perhaps even more exciting is if we have a dimensional variable or a continuous variable, we can build some kind of regression model in our training set that's relating some kind of brain measure to this phenotypic variable. This could be a symptom score, or performance on a cognitive task, etc. And once we've built this model in our training set, we can test it on a brand new individual from the test set that we've never seen before, and we can ask, can we generate an accurate prediction for where this person falls on the phenotypic spectrum? And if we can do that, that's very powerful. So to give an example of this, our lab was interested a couple of years back in seeing whether we could predict the individual trait of fluid intelligence from someone's resting state functional connectivity. So we turned to the Human Connectome project data and found that using a very simple machine learning technique, we could actually build a model on a subset of the subjects in the data set that related strength in certain functional connections to fluid intelligence. And then we could test that model on a brand new subject's functional connectivity profile. So now for each subject in the data set, we have their actual score as well as the score that our model predicted for them, and we can plot these against each other. And if our model is doing a good job, we would expect a positive correlation between these two. And that's in fact what we see. So now we're able to say not just that resting state connectivity correlates with fluid intelligence, but that resting state connectivity actually predicts fluid intelligence in someone we've never seen before. And these two terms, correlates with versus predicts, get thrown around interchangeably sometimes, but they're actually quite different. Excuse me. And prediction is a much harder problem than correlation, but it's a much more important problem, again, if we want our findings to have practical utility someday. And what's even more comforting is that other groups have actually discovered this same predictive relationship also using HCP data. And here are some references here. So it should be said that these are not direct replications, and there's actually a considerable amount of variability in the approaches taken by these three papers. And, the connectivity features that we're each using are slightly different. The regression models are different. But that's actually a really good thing because it's giving us these nice converging findings that make us trust the results even more. So the fact that there are correlates of fluid intelligence in people's resting state connectivity. And that's one of the nice things of using large open data sets is that often there's multiple groups sort of working in parallel, and it's not so much duplication of efforts, but rather it's this nice convergence that if we're all finding similar things, even with our slightly different methodological choices, we can all have a lot more confidence in those results. Okay, so that's just one example to say that we can learn some interesting things just by looking at a single large open data set on its own, but we can learn some really interesting things by combining large open data sets with smaller data sets that we've collected in our own lab. So I don't think many of us would argue that large open data sets should supersede smaller focus data sets that we're collecting in labs around the country and have been for many years. I think rather the large open data sets can be a really nice complement to the smaller lab-grown data sets. And there's sort of two ways that this can work. So one, let's say that you design an experiment and collect data to answer a relatively small small focused research question, and that's, typically what the majority of fMRI labs have been doing over the last couple of decades. So if you've collected this small focused data set, you probably have a hypothesis that's kind of driving your analysis. That's why you set up your experiment that way. So you probably conduct this analysis on your data and you end up with some result. Hopefully it's something cool and interesting. If this were 10 or 12 years ago, you'd probably just stop here and publish your findings. But with the availability of large open data sets, it opens the door for you to take this a step further and ask if there's sort of maybe just part of this result, or maybe the whole thing, that you could take and apply to a large open data set to ask this question about generalizability. So maybe the large open data set doesn't contain exactly your paradigm, but that's actually even better. So is there a part of your result that you'd expect to be able to generalize, and can you test this with the help of data that's already been collected and is freely available? Another way that you can combine a large open data set with a smaller lab-grown data set is to sort of work the other way, from large to small. So say that you're just starting grad school, or you're just starting your postdoc, and you just kind of want to play around, and you download a large open data set. The way that most of these are set up is it's going to be pretty exploratory. So you run some kind of exploratory analysis and you get an interesting result. This was an exploratory study, but because you have a lot of subjects going into the analysis, you tend to trust it. But maybe that brings up some really interesting questions and you want to dive further. And so maybe you use that result to inspire the design of a new, smaller, focused data set that you can use to again, take this a step further and ask a more subtle or more mechanistic question. Can we learn more about what's driving this result that we've observed in this large open data set? And I'll give an example for each of these of how this might work. So starting out from small to large, this is a real-life example from a colleague of mine, Monica Rosenberg. So in grad school, Monica was really interested in studying the neural basis of attention and what allows people to sustain their attention for long periods of time. And if you made it this far in the course, this is probably something that's also of interest to you. So what Monica did was she gave subjects, healthy subjects at Yale, this challenging gradual continuous performance task. And you're seeing the task right now in real time. So subjects are lying in the scanner, they're looking at these images, and their task is just to press a button every time they see a city scene and not press when they see a mountain. Sounds pretty easy, but it's tough to keep your attention to these stimuli for the tens of minutes that she was having people do this task. So there was a lot of variability in how people performed on the task, and she wanted to see if she could predict how people would perform from their functional connectivity. So again, Monica gave this task to 25 healthy adults at Yale, and even within this smaller data set, she was able to show, using leave one subject out cross-validation, that she could predict someone's performance on this task just from their resting state functional connectivity. So that's what you're looking at here, is a positive correlation between the behavior that she observed people to have and the behavior that the model predicted, showing that we can in fact pick up on this type of individual differences in attention. But Monica wanted to take this a step further. So rather than just stop here, we were sort of asking ourselves, well, have we just discovered something that's very specific to adults in New Haven's ability to perform this weird city mountain task in the lab, or have we discovered something that's more generalizable about the way that attention is reflected in the brain? And in order to get at this, with Monica, we tried doing cross data set prediction. So we obtained a bunch of data from the ADHD 200 consortium. This was resting state data from 113 children that was acquired at Peking University in China. So now we're changing up a lot of different things. We have a different subject population, obviously a different scanner, different site, different MR protocol, etc. But if our model is really picking up on something that's generalizable about attention, we should be able to apply what we learned from the Yale dataset to this ADHD 200 dataset. And so when we tried running the model that we developed on the Yale adults to these children from Beijing, and I should say here that what we're predicting here So we didn't actually have the children do the City Mountain task. Instead, our measure of attention was this ADHD rating scale, where higher scores indicate more severe symptoms of attention deficit hyperactivity disorder. So that was our score for the kids, and we had their resting state data. And if our model is working, we would actually expect a negative correlation between what we predict, how we predict that they would hypothetically perform on the City Mountain task, and their ADHD score. So people that have more severe symptoms of ADHD, our model should predict that they would perform worse on the task just by feeding it their functional connectivity. And that's in fact what we found. And that was really compelling. So, it's not working perfectly, but the fact that this generalizes across two extremely different data sets gives us a lot of hope that what we're seeing here is not just unique to the specific subject population or the specific task, but it's something more low-level true about the way that attention is reflected in the brain. Okay, so another way that we can use large open data sets to sort of inform and inspire the work that we do in the lab is to work from large to small. And here is an example of that. So turning back to our analysis on the Human Connectome project from a few slides ago, here's sort of an expansion of that analysis. So before I showed you using resting state connectivity to predict fluid intelligence, this GF variable here. But the nice thing about the Human Connectome project is they have not only resting state data, but data from lots of different cognitive tasks, and that's what's along the y-axis here. So we are asking the question, which of these task conditions does the best job of predicting cognitive behaviors? And we've expanded to a few other cognitive measures here. And there's a lot going on here, but what I'd like you to take away from this is that what we noticed when we expanded this analysis is that the two resting state scans are actually some of the worst predictors of cognitive behavior. They're still significant predictors, but they do a worse job of predicting individual differences compared to things like these tasks up here. So this is the working memory task, the language task, the gambling task, the social task. And when we thought about this a little bit more, we thought that maybe these tasks are actually serving to enhance or bring out individual variation in the functional connections that we care about. And this was interesting and not obvious from the start to us. It was something that we had to discover empirically by working with this large open data set. And when we thought about why this might be the case, these are tasks that are challenging. A couple of them are sort of more naturalistic in nature, interactive. And so maybe by having this task on board, again, we're drawing out more individual differences in places that we care about. But this, again, was an exploratory analysis, so that can only take us so far. But the idea now is we're using this result to inform a new study where we're asking the question, can we design a condition specifically to draw out individual variation in a behavior that we care about? And you can think of this as kind of like a stress test for the brain. So can we specifically probe certain systems? And this has now led us to create a novel naturalistic task paradigm that's trying to capture the features of some of those most successful tasks. So it's naturalistic, it's engaging, it's challenging, and the idea, and now we've given this task now to about 20 healthy subjects, is that everyone's going to listen to this story, undergo this paradigm, and they will arrive at different interpretations of the story, and we will try to predict the individual differences in these interpretations from brain activity while they were listening. But it was really that exploratory result that we obtained in the HCP data showing the superiority of certain tasks over rest that inspired us to design this new study. So that's a way that you could work sort of from large to small. Okay, so let's say you're convinced, you're ready to start working with large open data sets. Here's a couple, just a very sort of broad overview of the pros and cons. Some of the pros are pretty obvious. The big one is that they're already collected, so that's a big time saver for you as a researcher. They're very large, which again buys us a lot of the statistical power that we've been talking about. They typically use tried and true paradigms. I'll be careful when I say too much about this. Things like the Human Connectome project have both rest and task. The tasks tend to be pretty well studied things like an NBAC task for working memory, an emotional face processing for the emotion task, things like that. So they're kind of operating with paradigms that have been used over many years and we know a lot about. We can argue over resting state data, but One thing about it is that it's very convenient and easy to standardize both the acquisition and the analysis. So that is a nice feature that makes it lend itself quite well to being included in large open data sets. And usually, if you're working with a large open data set, it's already pretty well documented. So that saves you a lot of hassle. If you want to be a good citizen of the science world and document your data sets, well, if you're working with an open one, someone's already done that for you. So that's nice. But there's no such thing as a free lunch. So working with large open data sets also come with some cons that are sort of the direct oppositions of some of these pros. So pro, it's already collected, but con, you have less control, right? So you're sort of limited to what the original experimenters have chosen to do. It is quite large, but that doesn't mean that you can always use all of the data. So data quality can be an issue, and it's sometimes even more challenging to check for data quality when you have such a large number of subjects. Because they are so large and designed for exploratory analysis, sometimes you can get a less creative experimental design. So you're sort of working with the lowest common denominator of things that have been done, and there's maybe less room for super novel or sort of untested paradigms. And even if it's well documented, the fact that many labs are working from the same data set means that if there's an error in it, these errors can propagate across multiple studies and analyses, and that can be a big problem. So these are not, these are just things to be aware of when you start working with these types of data sets. So one thing that you can do to help mitigate some of these disadvantages, obviously, these things should go without saying, but it's always good to have a reminder. So always read the documentation very carefully. Don't make assumptions about what has or hasn't been done to the data, especially in the fMRI world where our data undergoes so many different pre-processing steps before we're ready to start looking at it. Definitely don't assume that the data has or hasn't been pre-processed or which steps have been done and which haven't. It's your responsibility to read the documentation and know what's been done or not. Different data sets have different philosophies when it comes to releasing sort of poorer quality data. Some data sets will clean first and only release good subjects, but it's becoming increasingly common to actually release all of the data because that way people that are actually interested in developing better data cleaning up techniques can test it out on some lower quality data, but that means that you should do your own quality checks early and often with the data that you're working with. And as the ends increase, it becomes less practical to do, to open things up and visually check and manually inspect every single subject, but there's tools out there that can help with this. So one really cool new one is MRI-QC, which does automatic prediction of quality and visual reporting of MRI scans. You can check that out at mriqc.org. Don't be afraid necessarily to chop down the sample to, a high quality subset of data. And one thing that we've had experience with in our lab is chopping down the sample to sort of try to mitigate correlations between behaviors and phenotypes that we're interested in and head motion. So many of you are probably aware that head motion poses a pretty big confound, especially for functional connectivity analyses, and unfortunately, many of the behaviors that we care about are correlated with head motion. And here's a nice paper that looked at this in the HCP data, finding that people that score higher on a lot of the cognitive measures tend to move less in the scanner, whereas people with a higher BMI or a lot of somatic problems tended to move more. This is not surprising. We also know that there's big age effects on this. So kids, both young kids and older adults tend to move more than healthy young adults. And these effects, may not be gigantic, but they actually, they reach a pretty strong significance level when you're working with hundreds or thousands of subjects. So you really need to be aware of these types of confounds in these data sets. And one way that you can mitigate this is by chopping down the sample. So here's just another harking back to our prediction of fluid intelligence from resting state connectivity. We actually did have to chop down the sample a little bit. So we observed that within this sample of 126, and again, this was an earlier HCP release, we observed that the mean frame-to-frame motion was negatively correlated with the observed fluid intelligence, such that people that scored higher on the fluid intelligence moved less. This wasn't a massively strong correlation, but it was enough to make us worried, and we didn't want to have this confound. So before your analysis, you can check for this correlation. You can't control for it if you don't check for it. what we ended up doing was we were able to exclude just some extreme subjects that were sort of driving this correlation down. So once we excluded just these 8 particularly high motion subjects, we were pretty satisfied that we had removed this systematic correlation. And the nice thing about these very large N's is, if you only have 20 subjects and you chop out 8, well, that's almost half your sample size. But if you have hundreds of subjects and you chop out 8 or 20 or even 50, it still leaves you with a lot of data to work with. And you want to be careful. You don't want to handpick or cherry pick subjects, but it does give you the opportunity to work with a high quality data set that's still quite large, even if you chop out some subjects. And then, of course, you can always, if you're doing some type of cross-validation or behavioral prediction, you can always do a double check for this type of confound by also, after you do your brain behavior analysis, make sure that your predicted behaviors are not correlated with motion. So here we're proving that our observed behavior is not correlated with motion, but we can also check that our model is not just picking up on variance associated with head motion by correlating the by correlating the predicted behaviors with mean frame to frame motion, and we don't see a correlation here either. So that gives us some confidence that motion is not a confound in this study. But these are just the steps that you should try to take on your data depending on your research question. And before I wrap up, I just want to mention a sort of different kind, what I'm calling a different kind of large open data set. We tend to think, and I've been talking certainly in these terms throughout this talk, about a large open data set, meaning a data set containing many, many individuals, but who are relatively sparsely sampled. So maybe you have a couple scans on each person. If you're lucky, you have some behavioral metrics, but it's typically from one time point. So many individuals, few samples per individual. And there's been a recent movement that I think is very commendable to complement these types of data sets with another type of data set, which is that people are collecting lots and lots of data on few individuals. And so some examples of this would be the MyConnectome data from Russ Poldrack. So this was Russ scanning himself every Tuesday and Thursday morning for 18 months. There's about 84 rest new state scans, 40 task scans. He did blood draws on mood surveys, etc. So this is a very densely sampled single individual. And inspired by this, the Midnight Scan Club has just released their data set, which contains 10 individuals worth of data with hours and hours of both rest and task on each person. So this is a different way of thinking about big data. Instead of getting a little bit of data from a lot of people, you're getting a lot of data on just a few people. And this could be a really nice complement. So you can imagine that if you have a really high signal to noise, even if you have a smaller population, you could try to build a model on these really well characterized individuals, and that may be a better model that you could then test on a larger data set where maybe you have a little bit less data but you have a lot more subjects, and thinking about ways to sort of use these different types of data sets in ways that complement one another. So to start to wrap up here, hopefully you've gotten some of these take-home messages that large open data sets can be a very powerful tool for discovery. But it's on you to know the strengths and weaknesses of your data set. I don't think that large open data sets should ever replace sort of smaller, more hypothesis-driven focused studies, but I think that there's lots of opportunities for these types of studies to complement one another. And if you're thinking about brain hack projects, then large open data sets can be a really great starting point. Since a lot of people are already familiar with them, they provide sort of, a common denominator for everyone to start working with them. A lot of the documentation's already been done for you. It's easy to collaborate remotely and across sites, so they present a really exciting opportunity for discovery in brain science these days. So I hope that you've enjoyed this lecture, and best of luck with the rest of the course.